Welcome to Advanced Learning Tutoring. This is the biology playlist. Please like this video and subscribe to our channel. Cell structure. Plant and animal cells are both examples of eukaryotic cells. These cells have a cell membrane, cytoplasm, and a nucleus that contains the genetic material, the DNA. Eukaryotic cells are larger and more complex in structure than prokaryotic cells. An example of a prokaryotic cell is a bacterial cell. These are much smaller in comparison. They have a cytoplasm, a cell membrane, and are surrounded by a cell wall. The genetic material is not enclosed in a nucleus. The DNA is found as strands. There is a single loop of DNA, which is known as a plasmid. Animal cells contain a cell membrane, ribosomes, a nucleus, cytoplasm, mitochondria. Plant cells have a cell wall, a cell membrane, ribosomes, a nucleus, vacuole, cytoplasm, mitochondria, and chloroplasts. The nucleus controls the cell as it contains the DNA. The cell membrane allows substances in and out of the cell. Substances include oxygen, carbon dioxide, water, glucose, and these substances move into and out of the cell via the cell membrane through processes such as diffusion, osmosis, and active transport. Ribosomes are the site of protein synthesis. This is where proteins are made. Examples of proteins are enzymes and hormones. The cytoplasm is a jelly-like substance. It's the site of various chemical reactions. Mitochondria is the site of respiration. All living things respire. Respiration is the process of releasing energy. The chloroplasts, which are only found in plant cells or those that photosynthesize, contain a green pigment called chlorophyll. This is where photosynthesis occurs. The cell wall is made of a substance called cellulose. Cellulose is a protein and it supports the cell. The vacuole is filled with cell sap. Required Practical 1, using microscopes. First, we need to understand the difference between a light microscope and an electron microscope. A light microscope is one in which you would use at school, and we are able to observe cells. Using a light microscope, you can also observe the nucleus of a cell. They have an eyepiece lens, which usually magnifies objects times 10 magnification, a coarse focus wheel and a fine focus wheel, both used to focus your image, an objective lens, which is on the dial, you can rotate to higher magnifications. When using a light microscope, you start on the lowest magnification and then rotate to increase magnification. The stage holds the glass slide with the object on it. You use a lamp to shine a light at the mirror, which reflects up into the lens. An electron microscope has a higher magnification and resolution than a light microscope. An electron microscope can be used to identify subcellular structures, for example, mitochondria. You can calculate magnification by dividing the size of the image by the actual size of the object. Magnification has no units. Therefore, the measurement of the size of the image and the actual size of the object need to be in the same units, for example, millimeters. Due to cells being so small, you can normally measure the actual size of a cell in the micrometer scale. 1 micrometer is 1 times 10 to the minus 6 of a meter. 1 millimeter is 1 times 10 to the minus 3 of a meter. We can use this to convert between micrometers and millimeters. 1 millimeter is equal to 1,000 micrometers. Therefore, to convert between millimeters and micrometers, you multiply the millimeter reading by 1,000 to get your answer into micrometers. Cell specialisation and differentiation. Cell differentiation. Animal cells differentiate at an early stage. Embryonic cells become specialised. Plant cells, however, are able to differentiate throughout life. An example of this is that you can cut down a plant and it will regrow. However, in an animal, if you were to amputate a leg, for example, it would not regrow. And this is because the cells in an animal will not differentiate throughout the life. Specialised cells are those that have adapted their structures to their function. Understanding what a stem cell is an undifferentiated cell. In an animal, your stem cells are either embryonic stem cells or stem cells found in bone marrow. Embryonic stem cells are formed when a sperm and egg cell fertilise. This fertilised egg cell then divides by mitosis. You then have a group of identical cells known as an embryo, and these are able to differentiate into any type of cell. Specialised cells in plant cells. Phloem. The phloem is a tissue made up of many phloem cells. Dissolved sugars and amino acids can be transported both up and down the stem in the phloem. Companion cells adjacent to the sieve tubes provide energy required to transport substances in the phloem. Root hair cells found in the roots of plants are underground. The root hair cell has a large surface area to provide contact with soil water. It has thin walls so as not to restrict the movement of water. It does not contain chloroplasts, as these cells are found underground. Chloroplasts are the site of photosynthesis, which is where the chloroplasts absorb light energy to react to produce glucose. As these cells are found underground, no light can reach the root hair cells. The xylem is a tissue made up of xylem cells. There are no top and bottom walls between xylem vessels. It is a hollow tube, meaning there is a continuous column of water running through them. Their walls become thickened and woody, and therefore they support the plant. Culturing microorganisms, a required practical for triple science students only. First, we understand how bacteria multiply. The process is known as binary fission. Bacteria multiply by simple cell division, similar to mitosis, dividing into 2 daughter cells as often as once every 20 minutes, providing they have enough nutrients and a suitable temperature. Bacteria can be grown in a nutrient broth solution or as colonies on an agar gel plate. Uncontaminated cultures of microorganisms are required for investigating the action of disinfectants and antibiotics on a sterilised agar plate. The method: Petri dishes, nutrient agar jelly and other culture media must be sterilised. The inoculating loops used to transfer microorganisms must be sterilised. This is done by passing the metal loop through a Bunsen burner flame. The lid of the Petri dish is sealed with sticky tape to stop microorganisms from the air getting in and contaminating the culture. You measure the zone around the antibiotic where there is no bacterial growth. The larger the zone of inhibition, where no bacteria is growing, the more effective the antibiotic. Cell division. We're looking at the process of mitosis. Mitosis is a type of cell division in which cells divide for the process of growth and repair. The nucleus of a cell contains chromosomes, made up of DNA molecules. Each chromosome carries a large number of genes. In body cells, the chromosomes are normally found as pairs. During mitosis, cells divide in a series of stages called the cell cycle. Two identical daughter cells are formed from one parent cell, and these are known as clones. The first stage of the cell cycle is a growth stage. The DNA replicates to form two copies of each chromosome. The nuclear membrane breaks down and the chromosomes line up across the centre of the cell. One set of the chromosomes is pulled to each end of the cell and the nucleus divides. The cytoplasm and cell membrane divide to form two identical cells. They have the same number of chromosomes, mitochondria and ribosomes as the parent cell. Stem cells. A stem cell is an undifferentiated cell of an organism which is capable of giving rise to many more cells of the same type, and from which certain other cells can arise from differentiation. Describe the function of stem cells in embryos, in adult animals, and in the meristem tissue of plants. Stem cells from human embryos can be cloned and made to differentiate into most different types of human cells. This is useful to treat disorders such as diabetes and paralysis. Stem cells from adult bone marrow can form many types of cells, including blood cells. Meristem tissue in plants, which are the shoots and the roots, can differentiate into any type of plant cell throughout the life of the plant. In therapeutic cloning, an embryo is produced with the same genes as the patient. Describe the advantages and disadvantages of therapeutic cloning. The advantages include stem cells from the embryo are not rejected by the patient's body, as the DNA is the same. Stem cells from meristem tissue in plants can be used to produce clones. This enables rare species to be cloned to protect from extinction. Crop plants with special features, such as disease resistance, can be cloned to produce large numbers of identical plants for farmers. Disadvantages, however, is that the use of stem cells has potential risks such as transfer of viral infection, and some people have ethical or religious objections to using stem cells from embryos. Transport in cells. Diffusion. Substances may move into and out of cells across the cell membrane via diffusion. Diffusion is the spreading out of particles of any substance in solution or particles of a gas, resulting in a net movement from an area of higher concentration to an area of lower concentration. What substances go into and out of cells? Oxygen and carbon dioxide in gas exchange in the lungs, and the waste product of urea from cells into the blood plasma for excretion in the kidney. What factors affect the rate of diffusion? The difference in the concentrations, i.e. the concentration gradient. If there is a larger difference, in concentrations, there will be a greater net movement in substances. Temperature. The higher the temperature, the faster the rate of diffusion. And the surface area. The larger surface area increases the rate of diffusion. A single-celled organism has a relatively large surface area to volume ratio. This allows sufficient transport of molecules into and out of the cell to meet the needs of the organism, without the need of specialised exchange surfaces. This is different to a multicellular organism, which has a much lower surface area to volume ratio. Multicellular organisms, for example humans, require specialised exchange surfaces such as the alveoli in the lungs and the villi in the small intestine. Transport in cells. Adaptations for diffusion. Explain how the small intestine and lungs in mammals, the gills in fish, and roots and leaves in plants are adapted for exchanging materials. Each of these exchange surfaces has very similar adaptations. All have a large surface area, which increases the rate of diffusion, a membrane that is thin to provide a short diffusion path. This is commonly known as a thin wall. In animals, having an efficient blood supply, and in animals for gas exchange, being ventilated, i.e. the gills. The alveoli in the lungs are surrounded by a capillary network, which provides each alveoli with a good blood supply, ensuring that gas is exchanged directly into and out of the bloodstream. They have very thin walls, providing that short diffusion pathway, and the alveoli are shaped to increase the surface area. The villi in the small intestine are finger-like projections that poke out into the small intestine. This increases the surface area, allowing for a faster rate of diffusion of nutrients into the bloodstream. They have a thin wall for a short diffusion pathway, and each villi has a capillary running through it, meaning nutrients from digestion are able to be absorbed straight into the bloodstream. Osmosis is the movement of water from an area of high concentration to an area of low concentration through a partially permeable membrane. Animal cells also take in and lose water by osmosis. They do not have a cell wall, so will change in size and shape when put into solutions that are at different concentration to the cell contents. For example, red blood cells could lose water and shrink, or gain water, swell and burst in a more dilute solution. Dilute solutions contain more water, therefore having a higher concentration of water. Plant cells, in pure water, the cell contents pushes against the cell wall and the cell becomes turgid. This is because in pure water, the solution has a higher concentration of water than the cell, so water moves into the cell via osmosis. In a more concentrated solution, i.e. a lower concentration of water, the cell contents loses water by osmosis. Water moves out of the plant cell into the solution. They shrink and pull away from the cell wall. The cell becomes flaccid, which is known as being plasmolysed. In a very concentrated solution, i.e. a very low concentration of water, the cell undergoes full plasmolysis as the cells lose more water. Osmosis required practical. Scientists are able to investigate the effects of osmosis in living cells. They do this by either observing the cells with a microscope, placed in solutions of different concentration, or measure changes in cylinders or discs of fresh potato or beetroot, or any other vegetable. The cylinders will have a larger mass than the discs, so scientists will have a larger measurement to work with. To investigate the effect of a range of sucrose solutions on the mass of potato cylinders. This can be done to determine the concentration of cell sap in potato cells. This may also be done with salt solutions to determine the concentration inside the cells. This is the method used. Add 30 centimetres cubed of a 0.8 mole per decimetre sugar solution to a boiling tube. Repeat step 1 with equal volumes of 0.6, 0.4, 0.2 and 0 mole per decimetre sugar solutions. To note at this point, the 0 mole per decimetre concentration of sugar solution has the highest concentration of water. It's pure water. Cut 5 cylinders of potato of equal size using a cork borer. It's important to note that each potato cylinder is of equal size to ensure that the surface area is the same, as we know that surface area affects the rate of movement into and out of cells. Weigh each potato cylinder and place one in each tube. Remove the potato cylinders from the solutions after 24 hours. Dry each potato cylinder with a paper towel. This removes any excess water from the potato that could affect the mass. Re-weigh each of the potato cylinders. You can calculate the percentage change in mass of each potato cylinder by finding the difference in the start and the end mass and dividing this by the initial mass of each potato. Multiply this value by 100 to get your percentage change. If there is a positive percentage change in mass, water has moved into the cell as the concentration of water inside the cell was lower than that of the surrounding solution. If the percentage change is negative, water has moved out of the cell as the concentration of water inside the cell was higher than that of the surrounding solution. You can plot these results on a graph. Where your line of best fit crosses the x-axis, where there is a 0% change in mass, that will identify the concentration of sugar solution inside the cell, as there will be no net movement of water when the concentrations are the same. Transport in cells, active transport. Active transport moves substances from a more dilute solution to a more concentrated solution, i.e. against the concentration gradient, from a low to high concentration. This requires energy from respiration. Active transport allows mineral ions to be absorbed into plant root hairs from a very dilute solution in the soil. Plants require ions for healthy growth. It also allows sugar molecules to be absorbed from lower concentrations in the gut into the blood, which has a higher sugar concentration. Sugar molecules are used for cell respiration.